I would like to thank very much Professor Ruby Pavankar, the president of Apaaaci, for this opportunity to talk more on epithelial barrier hypothesis and its mechanisms. And I would like to start my talk. The title of the second part of my talk is Epithelial Barrier Hypothesis Mechanisms. And I would like to talk about better understanding of epithelial barrier hypothesis for the increase of allergic and autoimmune diseases. So we understood in our research that epithelial barrier is defective in asthma, chronic rhinosinusitis, atopic dermatitis, colitis, bronchi, sinus, esophagus, intestine, skin, in all these material. And most of the human work are done by making biopsies in these tissues. You can show in the biopsies by RNA analysis or by RNA sequencing or staining with confocal microscopy or other methods. You can show that the barrier is defective in these diseases. We are nowadays using artificial three-dimensional skins, airway organoids, gut organoids, which are also giving the same results. So type 2 and type 1 response both open epithelial barriers. And we had a nice study with Paulina and Marcin, and we had this video about almost 10 years ago. And here IL-4 and IL-13 are used on air-liquid interfaces of bronchial epithelial cells. And you can see that the two walls of epithelial cells are not touching each other. There is 3 to 4 micrometer distance between the epithelial cells. And the epithelial barrier hypothesis says and also shows that the microbiome, which is floating on the surface of epithelium, is going between these defects. And together with microbiome, allergens are migrating to deeper tissues, and pollutants and toxins are doing this. So Terufumi Kubo published that there's defective bronchial epithelial barrier in asthma. This is a healthy bronchial mucosal surface, and we can see asthmatic bronchial mucosa, and you can see that the claudin staining one of the tight junction molecules, so one staining, they are both significantly damaged. Mikhail Zoyko published this also in the lab. Here we can see healthy sinus mucosa and occludin ZO1, two barrier molecules are stained, and this healthy sinus mucosa is showing a very intact sinus epithelial cell surface. And if there is chronic rhinosinusitis with nasal polyps, which is a disease which is affecting around 8% of European population, and you can see that there is a defect, and think that Staph aureus is sitting here, and itself is going deeper. It's all of it is toxins and enzymes are going deeper to make the chronic inflammation in the sinus mucosa with and without nasal polyps. So the barrier is very damaged. Likewise, we can see that in atopic dermatitis, and this is a normal skin, and there is filaggrin mutations which are continuously mentioned since 2007, and we have here claudin 7 staining of atopic dermatitis, and many claudins are important for the skin: claudin 1, 4, 7, and 8, and claudin 7 is very weakly stained, and it's also mRNA levels are significantly decreased. Another important cell that we have studied is innate lymphoid cells and type 2 innate lymphoid cells. Together with T helper 2 cells, they open epithelial barrier, particularly via IL-13. And the hypothesis is that if epithelial cells are activated and they release IL-33, IL-33 binds to ST2 on ILCs and type 2 ILCs. And what happens to the barrier? We approached this with a very elegant way, and we had RAC2 deficient mouse, RAC2 and common gamma chain deficient mouse, ROR-alpha staggered mouse, which is ILC2 deficient, and intranasal IL-13 was given, and human co-cultures and air-liquid interfaces were studied. So what we understood here is that IL-33 coming from the epithelial cells is activating type 2 innate lymphoid cells. IL-13 is going and damaging the epithelial barrier very significantly. There is a drainage response, expulsion response, which is a typical anti-parasite response, because larvae should be expulsed, as I have shown, so that they don't get adults in the lungs. And we, at the same time, allergens, toxins, pollutants, microbial products are all entering the tissues to cause a chronic inflammation. Now we come to collection of all these knowledge together with the studies of others, the epithelial barrier hypothesis. And this was published a couple of months ago. And we have here several important things. First of all, allergic and autoimmune diseases
showed a steep increase in the last several decades. This started in industrialized countries, but their prevalence continues to rise in developing countries. There is a defective epithelial barrier in allergic and autoimmune conditions, and it was demonstrated in humans and in mouse models very significantly. And this is together with dysbiotic microbiota that crosses the damaged barrier, and altogether this describes the epithelial barrier hypothesis. This hypothesis is explaining the development of around 2 billion patients, almost all allergic patients, and all autoimmune diseases have been demonstrated. And there's a group of neurodegenerative disease, and there is massive tissue inflammation or microinflammation in the affected tissues, and explains the epidemiological links to exposure to toxic substances. For example, in 1970s, 1980s, all of the workers of detergent companies who were inhaling the detergent before they had good measures and before they understood that their detergents are making people sick, they got all sick and they developed asthma. And at that time, there was a lot of link to healthcare workers for being exposed to detergents and also cleaning people. And this was a big link for epidemiological link to effects of detergents. And this hypothesis accepts hygiene and biodiversity hypothesis and links them to epithelial barrier defects in these diseases and microbial dysbiosis. It demonstrates ways of prevention, suggests future research direction, but it is very new to many of you, as I can imagine now. It needs a huge amount of work for public awareness and education, because we are continuously eating detergents because of professional dishwashers and household dishwashers. We are continuously inhaling detergents, and we are continuously eating food emulsifiers and other toxic material, which I mentioned, like the toothpaste, and also many other factors that affect the epithelial barrier. So if we look at the diseases, the first group of diseases are showing epithelial barrier defect and microbial dysbiosis in directly affected tissues. Here we have atopic dermatitis, asthma, chronic rhinosinusitis, allergic rhinitis, eosinophilic esophagitis, inflammatory bowel disease, and celiac. And in all of these diseases, there's epithelial barrier defect together with also microbial dysbiosis. Direct demonstration in humans done by us and others have been done a lot. In organoids, you can show these effects, and you can show in the biopsies together with microbial dysbiosis. The second group of diseases are autoimmune and metabolic diseases linked to gut barrier defect and microbial dysbiosis. We have here diabetes, obesity, fatty liver disease, autoimmune hepatitis, liver cirrhosis, rheumatoid arthritis, multiple sclerosis, systemic lupus erythematosus, ankylosing spondylitis. And in all these diseases, we are seeing here microbial dysbiosis, bacterial translocation, and most of these diseases are related to gut barrier weakness, but there's also lung barrier weakness or oral mucosal barrier weakness mentioned in these diseases. And in the mouse models, there's also microbial dysbiosis and gut barrier defect. Together with dysbiosis is a very common finding in all of these autoimmune diseases. And then we have the third group of diseases. These are chronic neuropsychiatric conditions linked to gut barrier defect and microbial dysbiosis. We can see microbial dysbiosis in all these, and there is necropsy studies which shows also gut barrier defect, but the evidence is relatively not as strong as autoimmunity or as strong as asthma in these diseases. It needs future work. That's what I can say at the moment. But there's enteric inflammation and microbial dysbiosis, for example, in Alzheimer's. Again, acute stress-induced gut barrier disruption, microbial dysbiosis, and translocation of bacteria in stress-related psychiatric diseases and chronic depression. So remember the expulsion response. The question is, what is expulse? What are we trying to expulse here in the bronchial mucosa or in sinus mucosa, in nasal mucosa? Here we have the epithelial cells. They are not touching each other, as we have seen from the confocal stainings. And the microbiome migrates deeper inside the epithelium. and gets in contact with basal epithelial cells. So the microbes and our normal microbiome, which should normally float above the epithelium, now goes deeper between the epithelial cells and also passes beneath the epithelial barrier. And this translocated microbiome starts a microinflammation there. And this is activating the immune system. And as I mentioned, the B cells see the staph aureus here and show anti-staph antibodies, which was almost not existing in normal humans or in asthma patients about 40 years ago. But nowadays, they have around 70% of asthmatitis and 90% of atopic dermatitis and chronic rhinosinusitis are showing Ig antibodies to staph aureus. So this is a very important thing. The other point is after translocation of the microbiome, there is a type 2 expulsion response. Now, the expulsion response, which is normally educated to be working for expulsion of parasites, mainly helminths, but nowadays the expulsion response is trying to expulse the microbes which are going deeper in the gut, in the lung, in the sinus. And here we have also colonized opportunistic pathogens. So the microbial dysbiosis starts and healthy microbes exchange their place with staph aureus, Moraxella, Haemophilus, and Pneumococcus. They become very dominant in asthma and chronic rhinosinusitis, and they become the main pathogens that the patients are not healing, and they are continuously being exposed to these microbes, and they are continuously showing a local tissue inflammation. And when they use, for example, inhaled steroids, then they, for a short time, they heal, but these diseases are never fully healing, and there is no cure. And these diseases last very long in life, and this is a very important point. And the other important aspect is circulating microinflammation and migration of inflamed cells and stem cells to distant organs. This is in autoimmunity, atopic march, autism, Parkinson's, Alzheimer's, and disease models. What's happening is that there's two exciting examples. One of them is Mario Zeiss and co-workers have shown from the gut that T cells are moving to the joints and making rheumatoid arthritis-like inflammation, and they have shown the same cells are going to the joints, and these are the T cells. Another important study was done in Stockholm, and about 2 years ago, in the outpatient clinic, the number of multiple sclerosis patients very significantly increased in January, together with increasing particulate matter. And particulate matter, which activated the dendritic cells and macrophages, educated the T cells to express CCR6 to migrate to brain. And this is an experimental finding, but in this model, the evidence and the association is quite strong. And if the multiple sclerosis patients are exposed to particulate matter, they show an increase in their disease and the severity of their disease. So here is a summary of epithelial barrier hypothesis. We have exposure to barrier damaging agents and opening of the barrier. There is colonization of opportunistic pathogens, and there is loss of commensals, microbial dysbiosis, and decreased biodiversity. So one of the major threats to humanity I mentioned is the decreased biodiversity, but this biodiversity is the whole world's nature. So certain species are getting lost, but we are also losing the commensals, healthy commensals, and they are getting exchanged with staph aureus or Moraxella, Haemophilus, and Pneumococcus. The opportunistic pathogens are colonizing. Translocation of microbiota to subepithelial areas makes the disease chronic because there is a small micro focus which makes a micro abscess under the epithelial cells, and these bacteria should not be there in the normal cases. Then an immune response to commensals and opportunistic pathogens starts, and inflammation in the epithelium and leading to chronicity is taking place, and defective epithelium cannot heal fully, cannot close the barrier, which we have seen in all our studies starting from 2010 in chronic rhinosinusitis, then in asthma, then in atopic dermatitis. So epithelial stem cells cannot make a full healthy epithelial barrier. So what are the future prospects of epithelial barrier hypothesis? I think there's a huge interest in the work because after the publication, I received around 400 emails as feedback and as a wish to work together. And this is going on. Actually, this work is being planned. I'll show you in the next slides. There's avoidance and dose control of all of the above mentioned products. I mentioned that they are decided as single product, but we are exposed to them together. So there's also synergistic effect to damage the barrier tissues. Development of safer and less toxic products. After our first detergent paper in 2016, many detergent companies started to develop less toxic products. That's a reality and that's a must for them. Discovery of biomarkers for the identification of barrier leaky subjects. This work is going on. Our skin impedance meter is working fine for the skin, but in the mucosas, we don't have very good biomarkers yet. Development of novel therapeutic approaches for tightening the tissue-specific barrier molecules is very important. Strengthening other components of the mucosal barrier, blocking bacterial translocation, avoiding the colonization of opportunistic pathogens, interventions through diet and microbiome, and many more novel approaches. As the European Academy of Allergic Immunology, we are writing now guidelines on environmental science in allergic diseases and asthma. Please write to us if you are interested in this. And also there is an epithelial cell biology working group that we are working together. And there's a lot of background and rationale to work on epithelial cell barrier. Epithelial cell is a very important innate immune cell, and it is initiating many of the inflammatory diseases. And as we have seen here shortly, it's also important for distant inflammatory diseases. So I will stop here by thanking our co-workers
who are currently working on the project. We have Milana Sokolowska, Arthur Rinaldi, Yasutaka Mitamura, Anja Haider, Juno Kim, Beate Rückert, Laçin Cevhertaş, Zeynep Sözener, İsmail Öğülür, Yağız Pat, Ozan Küçükkase, Duygu Yazıcı, and I'd like to thank Mübeccel. I would like to invite all of you to our WIRM, which will take place on the 6th of July in Davos. And this is our new institute. You are all invited to come here for research and education. Thank you for your attention.